Okay, so first the helicase unwinds the DNA, splitting it into two, and then the protein called the single-stranded binding protein attaches to stabilize the unwound DNA. So then next, primase adds RNA nucleotides to the exposed bases.
So DNA polymerase III will come in, and then with the different floating nucleotides, such as DGTP, it'll hydrolyze it, and then add the nucleotide, and then for DATP, it'll hydrolyze. Yes. The hydrolysis of the nucleoside triphosphates release energy and allows for DNA polymerase III to attach to the nucleotide bases. And then add it, and then it'll hydrolyze DGTP, and then add it.
To get rid of the primase, DNA polymerase I comes in, removing this, and then which removes the RNA primers, and then DCTP comes in, then it's hydrolyzed, it attaches, and DTTP also hydrolyzes, is attached onto there. So then this was the leading strand, so it'll produce continuously in the 5 to 3 direction. So now this is the lagging strand, which is going to go from the bottom up because it goes from the 5' to the 3' direction.
Because the DNA is made in a 5 to 3' direction, it causes DNA replication to occur in fragments in the lagging strand. This occurs due to the nature of DNA, which has an anti-parallel structure. Due to this anti-parallel nature, when DNA is split, one of the strands will be in a 3 to 5' direction. This 3 to 5' direction strand will become the lagging strand because it causes the DNA polymerase to add nucleotide bases in fragments because it is basically going from the back to the front in a 5 to 3' direction. Because of this, the lagging strand is made in fragments. In the case of the nucleotide bases, which is attached through the use of RNA primase and is currently attached to the parent strand, functions as the primer.
And they're produced in Okazaki fragments.
DNA polymerase I will come in and take off the RNA primase. It will then remove the primers by removing the nucleotides and adding nucleoside triphosphate, such as DATP, by hydrolyzing them, similar to the process done with the leading strand, which will then provide the energy and the adding of the nucleotide bases. In the case of this video, to save time, the nucleoside triphosphate has already been hydrolyzed or ripped. Similar to how the nucleotide bases were added previously, ligase will then come in to join the Okazaki fragments together. The ligase does this by taking different nucleoside triphosphates and hydrolyzing them. This allows for the energy and adding of nucleotide bases. Similarly to before, the nucleoside triphosphates have already been ripped or hydrolyzed to save time.